Okay, thank you very much. So I'm Takeshi Obayashi from Tohoku University. Can I use that? Okay. Oh, no, no, no. Yeah. So from Japan. Now my talk is about how to build gene co-expression resource based on the public databases, public transcriptome databases. Okay. So is this? No. Well, maybe this is okay. So yeah, so I'd like to first confirm what is gene co-expression. And of course, gene co-expression is similarity of expression profiles among the two genes in a genome. And yeah, so in fact, so I and my colleagues have been developing gene co-expression databases for plant species and animal species and microalgae for nearly 20 years. And the gene co-expression is, at least for me, it's an interesting topic because, so first, gene co-expression is gene-to-gene relationships, so we can easily draw gene network as well as protein-protein interaction network. But importantly, so the information reflects, so both of the protein-protein interaction network and the co-expression network reflect genome information, but the aspect is different. So protein-protein interaction network is derived from the coding region of the genome sequence. So if we get the set of the coding sequence in the genome, so that means we have the set of the protein structure in the genome or species, and hopefully, so we can predict protein-protein interaction network of a species. And similarly, so gene co-expression network is derived from the similarity of the expression profile, and the expression profile is encoded, fully encoded in the regulatory system or regulatory region in a species, or in the genome. So that means if we get the genome sequences, so we can determine co-expression network of a species. So that is our goal. So what is a good representation of co-expression network of a species? But the problem is, so gene expression is of course dependent on the condition. So for example, so if we calculate gene co-expression data based on the expression, for example, so specific to some condition or specific to, for example, the laboratory condition, so it's not the co-expression of a species, but condition-specific or laboratory condition-specific co-expression. So our goal is how to achieve co-expression condition-independent. So condition-independent co-expression of a species. That is a kind of mirror of expression, kind of mirror of genome sequence. So that is our goal. And I'd like to show several examples, actual examples. This one? Application mode. Here?
Yes. You use the down arrow?
No.
I think you need to prove that.
Okay. Yeah. Oh. Okay. I don't know. Sorry. Can I mirror the screen in this non-projector? No.
No, I think.
Okay. No.
Do we want to close this?
It's this? Yeah.
Which one is it?
Sorry. So, yeah. Okay. Yeah. Okay. Thank you. Okay, I'm sorry. So yeah, this is an actual example of gene co-expression. So there's 3 gene co-expression example here. Yeah. So, and of course, one gene and another gene and one gene and another gene and yeah, etc. And in the first and the second cases, this is positive gene co-expression. That means these two genes probably function together. And actually, in this case, in the first case, so both of the x-axis gene and y-axis gene function for photorespiration. So in these, for example, so, each protein indicates some condition over samples. And in these samples, the cell requires actually photorespiration function. So that is the reason why this samples or this cell actually upregulates these both of these genes. Now the next example is leucine biosynthesis, genes for leucine biosynthesis. So these genes function together for leucine biosynthesis. Now the third case is certainly negative correlation. So the interpretation of negative correlation is not so much straightforward, but we can easily hypothesize that the negative regulatory relationships between these two genes. Then actually, so these examples comes from the one large expression data set. And in the case of Arabidopsis here, in the case of Arabidopsis, so it comprises more than 20,000 genes and 20,000 genes and 10,000 samples. And this data are derived from the public databases. So in this case, NCBI-GEO or EBI-RA Express. And quite important point is this is the result of accumulation in public database. And it's not directly reflects the native condition of this species. So native condition is quite important concept to understand the genome information or gene co-expression of a species, because native environment selects the species and native environment determines the system of this species. So if we want to know or we want to determine co-expression of a species, so this sample collection should be responding to the native environment, but actually it's not. So the difference is quite important. And from the perspective of the native environment, so this accumulated data is somewhat biased or somewhat redundant or somewhat skewed. So the point is how to manage the redundancy, potential redundancy. And we applied principal component analysis to review the redundancy. So yeah, applied principal component analysis, and we get the principal components. And in this case, each principal component is can be regarded as a meta sample or meta condition. And importantly, so now this is independent. And so the upper three example is now shown in lower three example using the PC score rather than the original expression data. And this is much clearer. So again, so these two genes are co-expressed in even using the PC score. But now, importantly, this co-expression is supported by many independent meta conditions. So we can say that so this co-expression is always co-expressed in under various conditions. So we can say that this is stable co-expression. But in the middle case, so the original data is almost same. But in the next, in the center case, so this co-expression is heavily rely on one particular meta condition. So we can say that this is a kind of condition specific. So the condition is PC1, and PC1 is meta samples or meta condition here. But anyway, so this is a kind of condition specific co-expression. Now the third case is also interesting. So in the original data set, so third case is negative correlation, negative co-expression. But now, so we can easily quite straight linear correlation, negative correlation here. But this correlation is actually masked by first and the second principal components. And actually, so our observed data is a mixture of these three effects. So negative correlation and non-correlated factors. And apparently, so this original sample is somewhat redundant. And after applying the principal component analysis, so we can easily know the difference of the or hidden structure of the co-expression. And then, so we'd like to get co-expression of a species. So to do so, we'd like to distinguish these two cases. So it's not distinguished in the original data, but we can distinguish it using the PC scores. And this is stable co-expression and specific condition-specific co-expression. And probably, so we can imagine that, so stable co-expression imply more tight association of the function, because always these genes requires with each other for their function. And then, so we search, so what methodology is best to incorporate the difference between the stable co-expression and the condition-specific co-expression. Okay, so first, so the idea is when we simply apply Pearson correlation coefficient, we cannot distinguish these two cases. So both co-expression is quite large, so almost 0.8. But for example, so when we apply the Pearson correlation coefficient, so Spearman correlation coefficient, rank correlation coefficient. So Spearman correlation coefficient is resilient to the magnitude of the data, this data. So yeah, we can distinguish these two cases. And also ensemble calculation is good. So when ensemble calculation take into account the robustness of the relationships. And importantly, so now we usually, so bootstrapping is probably standard method to take account this ensemble for ensemble calculation. But we applied subsampling. This is important point because so usually, so bootstrapping samples same number of the data. So in this case, so here we plot top 1,000 principal components here, and in this case, so for example, when we applied bootstrapping, we pick up 1,000 principal component with replacement. Now that means, so for example, so this kind of super dominant condition or super dominant fact data always, almost always pick up in this in the 1,000 1,000 samples, and always this data hide the other data. So subsampling is best, better in this case. So for example, so we just pick up 50 principal components out of the 1,000 principal components. Now in such cases, so almost always we don't pick up this data, but we pick up the other data. And such partial data, partial for data composed of only 50 principal components can be regarded as condition-specific data. And such conditions, based on such condition-specific data, we can calculate the co-expression. And repeat and repeat and repeat. And the total of condition-specific data can be regarded condition-independent data or co-expression of a species. That is conceptually important point. And that is the reason why we adapted subsampling rather than bootstrapping. Now then, so we check the combination of some methodology using PCA or not, and using ensemble or not, and some index, co-expression index using Pearson correlation or Spearman correlation or its derivatives. And then, so we evaluate the performance based on the KEGG pathways. So if we get the good co-expression data, so we can distinguish gene pairs in the same pathway and gene pair from different pathways. And based on this score, so we evaluated some this combination of the methodology. And first, so simple Pearson's correlation coefficient is even better than the random level. Now, random level, sorry, is this dotted line, and 0.5 is the random line. And so naive PCC is not bad. But when we add some tricks, for example, so PCC MR is mutual rank conversion of PCC value, and mutual rank is actually the geometric mean of bidirectional ranks. And so more than 10 years ago, we proposed this method. And this methodology is actually, so we used this methodology in our databases until recently. And this methodology is better than the original Pearson correlation coefficient. But in fact, the combination proposed in this time using the PCA and ensemble and index is PCC transformed to the mutual rank is much better than the original one. And so now, we adapted to this methodology. An interesting alternative is just using the Spearman correlation coefficient. So this is not used ensemble. So this is computationally much faster, but slightly so the ensemble method is slightly better, but this is an important alternative. And then, so we evaluated this methodology using the other data. So I'd like to skip the details, but we checked this methodology, the superiority of this PCA ensemble calculation using the other species and both of the RNA-seq data and microarray data. So in every case, so this our proposed new method. So combining the PCA and the ensemble is better than the original methodologies. And I'd like to also pick up the specific cases. And when we compare, so PCA is quite easy to understand, and probably the key is ensemble method. And for I compared co-expression without subagging and with subagging, so subsampling aggregation. And in this case, so we plot these every gene pairs in one KEK pathway. So that KEK pathway is for valine, leucine, and isoleucine biosynthesis pathways. And this our ensemble strategy very specifically pick up some of the gene pairs. And actually, these some of the gene pairs make strong co-expression module. And quite interestingly, this module has distinct function. So neo-functionalization in only Arabidopsis. Even this is a homologous gene of valine, leucine, and isoleucine biosynthesis pathway genes. And actually, so this is proven by experiment, by knockout experiment by my colleagues based on our databases. That's good. So I'd like to summarize my talk. And so now, so we propose new co-expression calculation methodology combined with PCA and subsampling aggregation, subagging. And the key point is the PCA reveals the intrinsic structure of the co-expression. And we can now distinguish the stable co-expression and the condition-specific co-expression. And the subagging methodology incorporates these differences into a scalar, co-expression scalar. And this methodology shelterizes the minor component, not only the major component, but minor component, and reduces sampling biases. Now, these data are available for these species in our database. And finally, so this is our contributor. So currently, so I set up my new lab, and this is the lab I was previously belonged to. So thanks for listening.
Time for a couple questions.
Thank you for the interesting talk. Can you explain better your score to distinguish between genes that are in the same KEK pathway or in different KEK pathway?
Okay. So actually, so I have one additional slide. So the x-axis is our score, so co-expression data, and so when we make the two distribution, so that is gene co-expression data in different KEK pathway and co-expression data in the same KEK pathway. And this is basically the ROC curve, and we calculated the AOC. But the point is, we specifically focusing on the high low false positive area. So that means this right side, the difference of the right side is enhanced in this AUC calculation.
Okay. There was not an agreement between us. Okay, to clarify.